It is now my great pleasure to introduce our second next generation leader of the day, and this is Mark Harnett. Mark got his bachelor's degree from Reed College and then a PhD from the University of Texas, Austin. Now, you've heard a lot about the University of Texas, Austin, and their fine neuroscience programs. But I want to comment a little bit about Reed College. Reed College, a fine, small, liberal arts college just over 100 miles down the road. But there is something extraordinary about it. On a per capita basis, I believe it has produced the highest number of future neuroscience leaders. And not only just the number of them, but the quality of them. Professor Nobles, wouldn't you agree? Yes. Yes. Okay. Well, enough said about that. So during his sojourn at University of Texas, Austin, Mark worked on midbrain dopamine neurons, their physiology and their roles in addiction and motivated behavior. Then he moved on to a very productive postdoc at Genelia and worked there on dendritic computation. And now he's going to tell us about his current work, which focuses on individual neurons and their synapses and their roles in the biophysics of neural computation. Professor Harnett.
Thank you very much, Stephen. So that's a tremendous amount to live up to in an introduction. So hopefully I can do it justice. So I'd like to follow in Simona's footsteps and first of all say what a tremendous pleasure it is to be here with all of you, particularly to be elected to the Next Generation Leaders Council amongst such a great group of scientists. And to participate in the community here. This is huge for me, so thank you so much. It's a really fantastic opportunity. And secondly, I'd like to apologize, like Simona did, for my goofy voice. And I'm also going to blame my son. He's given me a sinus infection. So some of you in the audience may notice that this title seems a little familiar, and it's intentional. It's a specific homage to Christoph's book, The Biophysics of Computation, which was tremendously informative for me while I was a postdoc with Jeff. And this is a sort of publisher's summary of what the book is about. It was sort of my mission statement for what I wanted to explore. How is it that the specific biological features of neural networks that are very complicated and very nonlinear give rise to computation? And how is that different than the abstractions that we've found really useful in terms of building neural networks, simple neural networks, deep neural networks? So this is the tension that my laboratory tries to explore now. So I've been at MIT for just a year and a half, and I was sort of torn while thinking about this talk. Should I try to talk about a project that's already mostly done, that came from work previously, or should I talk about what we're actually doing in the lab? And I decided to go with the latter. So what I'll talk about is two specific projects we've actually made a little bit of progress on over the past year or so in my lab, one of which is very focused at the single synapse level on biophysics, and the other one is a sort of multi-scalar, as you all at the Allen love to say, kind of analysis of a specific circuit in the mouse brain. So what's the sort of goal of my laboratory? We're really interested in how single neurons give rise to computational power in mammalian circuits. We generally focus on the cortex of mice. And we look at single neurons, we look at their synapses, we look at how groups of synapses interact. And one kind of approach that we've used that's been really fruitful is thinking about what kinds of processing operations are linear and what kinds are nonlinear. So how is it that inputs come into single neurons or single dendritic branches and are processed? What kinds of mechanisms are responsible for doing that processing? Presynaptic, postsynaptic, et cetera. And what we've been really trying to do now, which started during my postdoc and is now a major focus in my laboratory, is not just do these things in the vast parameter space of a brain slice, but try to very tightly couple what we do mechanistically with what we see in a behaving animal. And I'll show you some of our behaving animal paradigms. And we use the whole sort of suite of modern systems neuroscience to probe this. Calcium imaging with a 2-photon, with a 1-photon microscope, optogenetics, new pharmacogenetic compounds, and then various forms of electrophysiology. And our goal is to try to pull out a couple of different levels of abstractions of some of this biological detail and figure out what are canonical operations being conducted at synapses, groups of synapses, and single neurons. Here sort of indicated by Bartlett-Mell's really beautiful multi-layer single neuron network, and try to eventually put these kinds of models into different types of neural networks. And then hopefully that will tell us a substantial amount about how cortical circuits work and may actually be spun off for sort of more general utility in the outside world for, say, a very efficient, power-efficient form of a neural network or something along those lines. So the first thing I'll tell you about briefly is a sort of corollary we noticed from a study I conducted with Jeff and a number of our colleagues at Janelia, and it's on the spine-neck resistance. So probably most of you, but maybe not all of you, know that dendritic spines are these little protuberances that come off of dendrites, and they're the site of the majority of excitatory synaptic connections in the brain. So vast majority of excitatory synapses are made onto these spines, and they have this very particular shape where here's the synapse, and it's connected to the parent dendrite by a very small neck. And this neck can act as an electrical resistor. And this has been a classical problem in neuroscience that was posed by Christoph and colleagues many years ago. And we tried to get at this problem a little bit more directly than anybody else had while I was with Jeff. And we used a combination of in vitro techniques, dual dendritic whole cell recording for electrophysiology, and then 2-photon calcium imaging and 2-photon glutamate uncaging to probe how this neck resistor influenced synaptic function and what was this actual value of this resistor. And what we came out with was a 500 megaohm number, which is substantially higher than I think most people had appreciated. And that is important. It has a bunch of functional consequences, one of which means that synaptic potentials inside the synapse are very large, like 20 millivolts or something, much larger than what you record at the soma, where most of these things are on the order of less than a millivolt. It also means that these very large potentials recruit nonlinear conductances that are present in spine heads. So we did some modeling experiments based on electrophysiology and showed that with spines, we got a lot more superlinear kinds of input transformations than without spines or with low neck resistances. So the corollary that came out of this was that if there's a great big resistor here, and it's sort of isolating the synaptic compartment away from the parent dendritic, it means that it would be almost impossible to voltage clamp a synapse. Voltage clamp is a particular kind of very quantitative electrophysiology technique that has been used for decades now to analyze synapses. All of our information, basically, about how synapses work in terms of the postsynaptic compartment and some of the presynaptic, right, comes from voltage clamp. And we were concerned that this very high neck resistor would have made those experiments basically uninterpretable. So we wanted to go ahead and directly test that. So a very talented graduate student in my lab started doing dual dendritic recordings from one of these rodent layer 5 neurons in an acute brain slice. So he's got 2 patch electrodes on here to validate how well he could voltage clamp the dendrite. And so here he's injecting current on one electrode and then recording the produced voltage on the other electrode. Right? So you just put some current in by Ohm's law, V equals IR, you produce a depolarization, a voltage. So now he turns on the voltage clamp. And what the voltage clamp is supposed to do is prevent any voltage excursion from occurring. It should just, as you pump current in, it should just record that current back to you without letting the voltage change. And if you do not set the voltage clamp perfectly under ideal conditions, which took us quite a while to find, you in fact recover far less of the current than you injected, and you allow a lot of the local voltage to change. So this is called escape. This is a huge problem for performing these measurements. We found conditions in which we could effectively voltage clamp this dendrite. And so now we wanted to use this paradigm and test, do we get voltage control inside a spine? Can we actually make an accurate measurement of the synaptic properties inside spines? So here we've got one electrode, and now we fill the cell with a structural indicator. We also fill it with a calcium indicator dye, in this case it's OGB, and we do a 2-photon imaging line scan through the spine head, and then we also uncage glutamate, very localized, just onto this spine head to activate the synaptic receptors as a proxy for the presynaptic terminal. We can increase this laser power and increase the amount of glutamate that's released, and we can build this nice curve. Here's the EPSP, right? The recorded postsynaptic depolarization at the electrode inside the dendrite. And then here are the resultant calcium signals inside the spine head that we're using as a proxy for depolarization. And in this case, these signals are produced by both NMDA receptors in the spine head and voltage-activated calcium channels. Now, we're going to switch to voltage clamp, which we know is controlling the voltage inside the dendrite from the previous set of experiments. And if it's controlling the voltage inside the spine head, we should see no calcium signals, right? The spine shouldn't be able to escape, right? It should just be completely suppressed. But instead, what we see is no distinguishable difference in the size of these voltage-activated calcium signals. It means that the spine voltage is basically totally uncontrolled by the voltage clamp, and it means that these currents that we're recovering are basically very, very, very severely underestimating what this should look like. So we can do this using either calcium channels or NMDA receptors. We've done a whole bunch of these now. They all basically do the same thing. It's very, very hard to voltage clamp a spine. We're measuring this now as well with voltage-sensitive dyes that we dialyze into the patch pipette rather than a calcium indicator for a more direct measurement. And so, okay, Mark, that's great. You've shown us that this is a huge problem. What are you going to do about it? So we've got a couple of approaches now based on those experiments I showed you for trying to reduce the effectiveness of the spine neck resistance and actually clamp the synapse effectively so that we can measure the properties, specific detailed properties under voltage clamp of the AMPA receptors, the NMDA receptors. No one's ever been able to show what's the AMPA to NMDA ratio at a single synapse in any sort of accurate manner, at least as far as I can tell. And I think if we can implement this effectively, we're going to learn a tremendous amount about the underlying synaptic physiology. And we can apply this to a number of different kinds of spiny synapses and different cell types. Okay, so now to switch gears completely to a sort of multi-scalar process and talk about retrosplenial cortex. So normally when I bring up the retrosplenial cortex, people's eyes go blank and they have no idea what I'm talking about. But since I've been here, virtually every person I've spoken to about the retrosplenial cortex has said, oh yeah, we've seen signals there. I work on this in humans, et cetera, et cetera, et cetera. So it's been very refreshing. So for those of you that don't know what the retrosplenial cortex is, it's an associational cortex. It's basically just medial to the visual cortical areas and just caudal to the anterior cingulate cortex. And it's important for spatial navigation and for spatial mnemonic activity. It is relatively poorly studied. It's highly interconnected with the visual cortex and with other areas of the brain associated with navigation and memory. So the reason that we're interested in it is that we're using it as a model system for an association cortex that's a little bit more tractable than something like the prefrontal cortex and something that has a fair bit of ethological relevance to what we can play with in a mouse in the laboratory. It's an area that is responsible for spatial navigation. We can very robustly explore and recruit that kind of system. And so our goal ultimately is to understand, right, how associative kinds of computations are produced in the cortex. That's sort of what we ultimately think cortex is doing. And so what RSC is thought to be responsible for is mediating the interaction between egocentric and allocentric spatial variables. And there's this great study from Doug Nitz's lab that's shown that single neurons in the RSC conjunctively code for both of these things at the same time. And so we have some specific hypotheses about how dendritic integration might be mediating this kind of process. But as I said, there's very little that's known about the RSC, so we sort of had to start at the bottom and build up. And so we've tried to go all the way from anatomy to physiology to population and computation. I'll just take you through a little bit of that. So we started with rabies-based monosynaptic tracing. In this case, this is an example from an RBP4 Cree animal, which labels layer 5 neurons in the A30 subdivision, which is the sort of dorsal bit of retrosplenial cortex, which is the easiest to image in the awake behaving animals. So that's where we've started. And you can see that there's reciprocal connections between this area A30 and this more medial part of the retrosplenial cortex. It's in the intrahemispheric division called A29, and there's also substantial intrahemispheric connections as well. It receives head direction input from the anterior dorsal nucleus of the thalamus. It also receives input not from the trisynaptic circuit of the hippocampus, but instead from the output region of the hippocampus, or one of the major output regions of the hippocampus, the dorsal subiculum. It also receives input from anterior cingulate cortex, but interestingly, none from the entorhinal cortices or perirhinal cortices. And there's a small, what looks like a small back projection from ventral orbitofrontal cortex. And there's some neat specifics in terms of which cell types from which lamina in these areas appear to connect to, in this case, layer 5 neurons versus layer 2-3 versus layer 6. So we're working on turning this more into, rather than a sort of messy ad hoc process where we just cut slices and put them on the confocal, into something more Allen-inspired with an actual pipeline that makes this a little bit more efficient with some of our collaborators at MIT doing whole brain imaging. Okay, so we know which neurons are coming into, say, a specific cell class in retrosplenial cortex, but where are those inputs synapsing on these elaborate dendritic trees? So we've adopted a technique invented by Carl Svoboda, S-Crackham, subcellular channelrhodopsin-assisted circuit mapping. And what we can do is then specifically activate inputs onto neurons in RSC from particular areas that we've identified with our rabies and figure out where they arrive in the dendritic tree. So here's an example from a layer 2-3 neuron in A30 in RSC. And the inputs that we've infected with channelrhodopsin here are from the anterior dorsal nucleus of the thalamus, and they're thought to convey head direction information to this circuitry. And what you can see is that there's no responses in the perisomatic domain when we run our photostimulation grid over these dendrites. We only see responses in the apical dendrites, and particularly strongly in the distal apical dendrites. And these are going to be filtered along this cable. They're already very large, so locally they've got to be very, very strong. And this sort of fits with what we see anatomically. These are just EYFP labels of these ADN inputs coming into retrosplenial cortex. You can see there's a sort of proximal apical band and then a very strong distal apical band. We know that there is some very interesting logic going on here beyond just anatomical overlap, because only about 20% of the neurons we've recorded thus far receive ADN input, at least as far as we can measure with this technique, despite the fact that those neurons are not, to us anyway, with our relatively crude techniques, are not separable. Electrophysiologically and sort of coarsely, as far as their morphology in the 2-photon, they don't look dramatically different. It's not like, oh, this very obvious neuron type gets ADN input and that one doesn't. So we're working on sort of clarifying this for these layer 2-3 neurons, and then also we're going to do this for all the other different inputs that come into RSC and all the different cell types. Okay, so now we know which cell types are getting what input, where that input's arriving in the dendritic tree, what kinds of mechanisms in these dendritic arbors are there to process that information, what kinds of filters are looking for particular patterns in the input. And so we switched to a technique that I spent a lot of time with in Jeff's lab, 2-photon glutamate uncaging, where we pick a dendritic domain on one of these neurons in a brain slice, and then we activate a whole ensemble of synapses in as physiologically a manner as possible. We're very careful and very paranoid about trying to make this very physiological. And we can play a bunch of inputs in at different spatial and temporal levels of clustering. And what we do is compare what we expect to get out of a particular branch as we add more inputs in. This is a sort of linear summation. And what we actually get out when we deliver the inputs clustered in space and time. And you can see this response here is much larger than what we would predict, and is associated with a very large local calcium signal inside the branch. So there's a huge superlinear component here for dendrites that are part of the basal arbor of either layer 5 or layer 2-3 retrosplenial cortical neurons. And this is sort of, I think, what you would predict given what we know from many other kinds of pyramidal neurons. Most of the branches in the perisomatic compartment of V1 layer 2-3 neurons, S1 layer 5 neurons, CA1 neurons, subiculum neurons, et cetera, all seem to exhibit this kind of nonlinearity. It is almost always mediated by the NMDA receptor, right? We put NMDA receptor blockers on, and this thing goes away, gets rid of the calcium signal, right? This is just the summary data. But what's fascinating is if you move to the apical oblique dendrites, which look indistinguishable as far as the spines, the density, all this other stuff, compared to the basal dendrites. They're not very far away. All of a sudden, you lose this superlinearity. What happens is a completely linear, completely pattern-insensitive integration that just eventually triggers the action potential at the soma. So there's no ability of these dendrites to sense pattern in their inputs that we can find. There's no contribution from NMDA receptors here. They just add up their inputs and then eventually trigger an action potential output. So we're really curious about what kinds of inputs arrive here. Why are these neurons set up like this, such that they're very specifically looking for pattern here and not here? We don't have the answer for that yet, but we're working on it. We're also wondering, what are the dynamics of the inputs that arrive there? Once we can anatomically figure them out, it would be really interesting to know what are they doing in terms of releasing their neurotransmitter. What does their activity look like? And so we turn to head-fixed rodent behavior, which by now I'm sure you're all quite familiar with. We have the animal running on a cylindrical treadmill while he's head-fixed. There's just a little head bar mounted on there. And then he's navigating down a virtual hallway. And so you can just see there's visual flow, there's motor output, and then he has a bunch of different kinds of contingencies in here, spatial contingencies that he has to decide for how to get reward. And in this particular case, we've got an optogenetic fiber in there into retrosplenial cortex, but we can also do two-photon calcium imaging through an implanted cranial window. Again, by now, sort of standard technique. We can image, hundreds of neurons in the retrosplenial cortex at the same time at reasonably high temporal resolution from different cell lines. We can genetically identify the neurons while the animals are doing this whole, a variety of these different kinds of head-fixed spatial behaviors. And we're starting to see some quite interesting cell types or response profiles come out of there. I'm not going to get into the specifics in the interest of time, but we can also image the other components in the RSC circuitry. Ooh, I was too slow to catch the movie. We can image axons and boutons arriving into the RSC from other regions. We just inject those other regions with GCAM. The contrast here is not perfect, sorry about that. But these are all axons and boutons arriving from the contralateral hemisphere of retrosplenial cortex. And now that we have some idea from our rabies experiments, what are the sources of input, we can go and inject into all of those regions as well. And we can, of course, image the dendrites. These are dendritic tufts in A30 of retrosplenial cortex. So it's layer 5 neurons, but we're imaging the dendrites in layer 1, right, which looks like the recipient area for head direction input in these particular neurons. And of course, the concern here, talking about head direction, is that animal's head fixed. There's no head direction input really occurring in an animal like that. And we were very concerned about how much of retrosplenial cortical function is dependent on free behavior rather than on head fixation. And so in a complementary line of investigation, we've developed what we think is a free behavior paradigm that requires retrosplenial cortex computations to accurately drive the behavior. So we call this the Thunderdome. It's a large circular arena with 16 ports around the outside of it. And those ports have beam breaks and LEDs and lick spouts. And we have a projector. Let's see if I can do this. Yeah, exactly. So one of my graduate students is obsessed with this idea. So I think probably for the sake of our lab's CAC accreditation, we should probably not have called it the Thunderdome. So here's an example of the behavior. Here's the mouse inside the chamber, and he's going to this port over here. He's going to this port over here. Once he successfully completes a trial, these light stimuli that are projected down onto the surface of the arena change. So he goes into a blackout period, and then lights come on in a different spatial orientation. And what the mouse has to do is realize where he is, see the spatial orientation of the lights, and then navigate correctly to the new reward ports. There's only 2 at a given time. And the relationship between the visual cues and the ports stays the same, but the animal's orientation in space and head direction changes very dramatically for how to effectively execute that behavior. So we've done a very crude, very uncontrolled experiment that I'll show you that at least sort of points to the fact that the retrosplenial cortex is involved in this kind of behavior. We just put a head-mounted LED onto the animal while he was executing this task in a VGAT animal and just basically turned off retrosplenial cortex while the animal was trying to do this behavior. And so in black is the response of the animal on interleave trials when there's no light. And you can see the animal does a pretty good job of picking the rewarded ports, right, compared to the unrewarded ports, these shoulder regions here. But when we turn the light on, accuracy of the animal in finding those ports, being guided by the light cues, goes down pretty substantially. OK, so I'll end with a project that I was really excited about when I found out that I was going to be able to have my own laboratory in Boston. I have always wanted to patch dendrites from humans. And you have beaten me to it. You have stolen my thunder. So I didn't think anyone else would be sort of goofy enough to try this. But I'm hoping now that there is some synergy here between some of the things that I'm interested in and some of the things the Alan's interested in, even though you're way ahead of me. So many of you are probably familiar with this recent paper from Idan Segev's lab. And one of the things I had thought was that based on the anatomy, there are really substantial differences in the electrotonic architecture of neurons from humans and rodents, right? The cortex is just much bigger. These dendrites have to span a much longer area. They're much thicker. There's more of them, right? There's more spine density. It's highly likely that there are specific biophysical mechanisms that mediate cellular level computations in humans that just don't exist or are very different in mice. And there are, of course, some substantial circuit differences as well. So one of these was just described by Idan, where they reported that the specific membrane capacitance of the membranes at the soma appear to be different in humans versus in mice. So that's already a good clue that there's going to be some pretty fascinating differences in dendritic integration, potentially as well in connectivity logic of the circuits. I'd love if we could manage to get some tissue from a frontal cortex or an anterior cingulate cortex that has a von Economo neuron in there. No one has ever been able to record from a von Economo neuron. We have no idea what their electrical properties look like. So this is something I'm really excited about doing, but as I'm sure many of you know, the red tape involved in getting approval to do this is ridiculous. So we've just gotten that all cleared out of the way, and we are hoping now to get our next surgical patient or our first surgical resection after Christmas and get started on answering some of these questions. So I think the achievement, if we'll call it that I'm the most proud of being a PI at MIT is being able to recruit this phenomenally talented team of individuals that I get to work with. And so I just want to acknowledge the fact that they're responsible for all of this work. And I'd like to say thank you again so much for allowing me to interact with the community, and I look forward to chatting with all of you. Thanks very much.
And thank you very much, Mark. So we're right on time, which means we have time for some questions. Raise your hand, and we'll try to get a microphone to you in sequence.
Yeah, 25 years ago, I guess, Francis Crick, had this paper on the twitching spine that depends on rapid modulation of the resistance of the spine using actin. Do you have any evidence for that, for any modulation of one particular spine resistance?
So I've never seen twitching on the order of, say, seconds. There's definitely evidence for shape changes on the order of minutes or maybe 30 seconds or something like that. So far, our data indicates that the morphological structure of the spine is not directly correlated with its electrotonic resistance. So it's something we haven't yet measured in terms of trying to induce structural plasticity and seeing a spine neck elongate or widen. And
but
then
I mean,
looking
you could have, for example, the spine apparatus move in or out dynamically, and unless you really look, you are likely to miss that, right?
Exactly. So that's the concern, is that how much of it is something that's beyond our resolution. But thus far, most of the spines seem to do basically the same thing. It's not like there's just some population of super low resistance spines and we're just picking some very specific spines that have this sort of bias.
But following up a little bit on Christoph's question, I mean, certainly the electron microscopic literature indicates that there's spines of many different shapes and sizes.
Indeed.
And of course, there is the question of dynamic modulation. So, I mean, how bold can you be in the generality of, the conclusion that you showed there?
I can't be that bold. The experiment sample number is relatively low because it's a pretty low throughput experiment that's a little technically challenging. And particularly under the 2-photon, you can't really be sure about spine geometry, right? You see this blob, right? It's not the same thing as super resolution or EM, of course. So we tried to stick with sort of average-looking spine. We didn't go for the ones that have these, really, really long necks that look more like Philipodium. We didn't go for the ones that look like little bumps on the dendrite. But as far as a sort of normal-looking spine, which, takes up a substantial fraction of the population, we think probably most of them do this. We haven't seen any counterexamples. We've done it in CA1 and in S1, layer 5 S1. And they both seem to behave very similarly. We haven't seen any massive differences there.
Okay, fascinating subject, but you talked about quite a bit, so maybe there's some other topics here. We'll talk about that one some more.
Okay.
Somewhat related to that question, I'm curious, given this high spine neck resistance, what you think the limitations are then for receptivity to backpropagating action potentials or activity of neighboring synapses on plasticity mechanisms in the spine head?
Fantastic question. So because of the electrotonic architecture here, things in the dendrite go back into the spine very effectively. The dendrite's a low impedance compartment, the spine is a high impedance compartment, and the main resistor in that assembly is across the spine head, not the spine neck. And so things go back. Action potentials, other dendritic spikes go into that spine very effectively. The drop, the filter happens for events initiated inside the spine dropping into the parent dendrite. The size of that drop depends on the parent dendritic impedance. Most of the experiments that I showed you here are larger dendrites where we can stick 2 electrodes on there. But we've gone and mapped into smaller dendrites as well. And you can see that this amplitude ratio from the synapse into the dendrite gets smaller and smaller and smaller as the dendritic impedance goes up. But as far as plasticity mechanisms, backpropagating action potentials, it all goes back to the synapse. It's a really sort of unidirectional filter, if you will.
Mike needs a mic.
Could you elaborate a bit on how the time courses and shapes of miniature synaptic currents are consistent with such a large resistance across the spine neck?
So what you're going to have in response to a mini recorded in voltage clamp is the spine voltage is going to depolarize substantially while you have AMPA-mediated current flow. So now that's going to decrease the overall amplitude of the current you're going to record, but it's also going to probably recruit some other conductances, be they calcium channels, NMDA receptors, BK channels, et cetera, some of which are going to increase the duration of that current, some of which are going to shorten the current. And so depending on the stoichiometry of what's actually in that spine, which can be very different from synapse to synapse, it's not entirely clear what you would get out. So it's hard for us to build a sort of quantitative hypothesis about what we expect, aside from voltage escape and distortion. And so we'd like to try to mitigate that to make a very accurate measurement.
We're so fascinated by spines. Isn't there anything else? Krista.
Yeah,
Stephen, there's somebody up here.
Oh, wait a minute. We have an, sorry.
Yeah.
Where?
On your left.
Right here, Steve.
Oh,
Hi.
Fire
Great
away.
talk. I have a question too, Christoph. So you've talked much about AMPA and NMDA receptors. What I had a question about was, have you thought about the interplay of inhibition, especially with the nonlinearities on these dendrites, and what do you think or if you have any ideas whether that has a special role in retrosplenial over other parts of cortex.
So that is a great question. That is a challenging topic to explore in terms of dendritic integration. It's been really challenging for us to try to figure out ways to quantitatively alter inhibition while we're probing excitatory integration. So there's evidence from Eli Nadivi and a number of other people now that there can be inhibitory synapses directly onto spine heads. It's a much larger fraction, I think, than people had appreciated previously. That's going to be a problem here as well, and is going to be even more of an issue if your voltage clamp is distorting things. I don't have a great answer for how you go about teasing apart the role of inhibition in this dendritic processing. We have a couple of little hints that there are some specific inhibitory subcircuits that may play a specific role in RSC. But I'm not sure I feel comfortable enough speculating on that. You know, give me 6 months or a year or so. But we're hoping there might be some neat inhibitory subcircuits in RSC.
Well, Mark, I feel compelled to just make the comment that things that are problems for us physiologists may be solutions for the brain.
Indeed. Indeed.
Okay, well, it's time to move on.
Thank you.